Welcome back to Unit 3, Lesson 6, Part 3 of the video. We're going to continue on and talk about what is recombinant DNA and how is it made. This is a really neat little trick we have for making recombinant DNA. So first, recombinant DNA is any DNA that is created by splicing, a fancy word for gluing together, splicing together DNA from two different sources. So, like two different organisms. Basically, it's designer DNA. And the way we do this is we take molecule A from organism A and molecule B from organism B. A real common example is we often take it from fluorescent jellyfish, and perhaps another one encoding for a gene from a mouse. And we attach the two together. And the neat trick is we use what's called an endonuclease, which is a restriction enzyme or an enzyme from bacteria that cuts DNA. Okay, acts like little scissors cutting up the DNA. Okay, and the really unique thing is each enzyme from bacteria cuts the DNA at very specific locations. Now, the reason bacteria has enzymes for cutting up DNA is they're very good at accidentally picking up DNA from around their environment, and sometimes that DNA could damage them. So if you believe in theory of evolution, they've evolved these enzymes to cut up foreign DNA so that they don't mess with their own DNA. But when they do this, they often cut not straight across the double strand, but leaving what's called a sticky end, an overhang. And if we take molecule A from organism A and cut it with the same enzyme that we cut molecule B with, it'll actually create these sticky ends that are complementary. That's why they're sticky. And then if you just mix the two DNAs together after they've been cut, they will complementary bind, and then we can seal them with the enzyme DNA ligase. Basically, glue them back together. And then we will have a recombinant DNA, a DNA that's made from two different things. And you're like, well, why would you want to stick DNA from two different things together? Well, we can do some really cool things with it. Sometimes we do it to create transgenic plants. Put a gene into a plant that is useful, for example, something that fights off disease or makes it resistant to pesticides. The other thing we use recombinant DNA is for medical treatment. So we've learned to put the gene for insulin, human insulin gene, into bacterial DNA, and basically the bacteria becomes a little insulin factory. It will make that protein for us, and then we just have a big vat of bacteria kicking out insulin, and we isolate the insulin back out of the bacteria, and we can give it to humans. Same with what's called factor 8 and 9 for people who suffer hemophilia. We use it to make human growth hormone and to make something called erythropoietin for treating anemia. As well, we use it for research. We can tag things with what's called the GFP protein. That's the green fluorescent protein. And this protein comes from, sorry I spelled fluorescent wrong, it's not flower, it's fluor. So this protein comes from jellyfish deep in the ocean, and scientists have taken the sequence for it and fused it to other proteins and made the proteins recombinant proteins that glow under UV light. And here is a picture of a GFP mouse. Now they're not making them so they're really cool at the club. You can take them to the club and they'll glow under UV light. They're actually making them so that we can see where proteins are expressed either in the whole mouse or within mouse cells. If it's at the cell membrane surface, you'll look at the cell under the microscope and only the outer edge of the cell will fluoresce. If it's in the nucleus, then only the nucleus will fluoresce. So it makes it easy for us to understand where the protein is working in the cell. As well, we can use it to target proteins to different locations, because proteins basically have amino acid address tags. And if you take that amino acid address tag, you take the code for it and you attach it to a protein that's normally in the cytosol, you can also send it to the membrane and see how that affects its function. So we actually use recombinant DNA for many different purposes. The last thing we're going to look at is how do gene therapies work. Gene therapies is therapies designed to replace mutated genes that cause diseases. So what we're trying to do is we're trying to deliver a wild type gene back into the cell so that it can resume making a functional protein. And we have no trouble now with all our DNA technology figuring out what mutations occur in genes, then creating the wild type gene back, and then at this point that's where the difficulty comes in. We now have the wild type gene, but we need to deliver it to the cell. And there's three main different ways we deliver the gene back into the cell. The first is using a retroviral vector. This is essentially a virus like HIV. HIV is great for targeting cells and getting its DNA into cells. So we take a virus, not HIV itself, but something like HIV, and we remove its own DNA so that it can't harm us, and we put in the DNA that we would like to deliver, and it will then target the cell, get inside and deliver its DNA into the cell. We can also use something like a adenoviral vector, and this is a virus like the cold, common cold. Different viruses are good for targeting different types of cells, so depending on what cell type you're trying to correct, you might choose a different type of viral vector. And the last one is a non-viral vector, where basically they make a little lipid coat containing the DNA, and that gets taken into the cell. The problem is the human body doesn't like foreigners, and when you add any of these foreign things, they often cause an immune response. So the trick is getting a vector that can deliver the DNA without creating an immune response. As well, once the DNA is in, the DNA needs to integrate in a stable fashion into the genome without actually disrupting the genome. So you don't want it just plopping down into the, inserting into the middle of some other gene and mutating that gene. Okay. So some things we've tried to use gene therapy for is treatment of SCIDs, which is severe combined immunodeficiency disorder. These you may have heard of, the people who are like bubble boys. Their immune system does not function properly because of a genetic defect, and it functions so poorly that if they're exposed to any illness, they're going to have a really hard time surviving, so they basically have to live in a bubble away from everyone. Now, it's to date has been met with various success. They've been successful at treating the combined immunodeficiency disease, so the immune system starts to function properly. Unfortunately, the way it's delivered into the system, it's delivered into blood cells, seems to create leukemias. So it's not integrating in a stable way, it's integrating in a way that disrupts cell replication. So now you've beat your immunodeficiency disease, but you have leukemia. There's only been a few trials, but in 2 patients, leukemia has been the result. Now, leukemia doesn't kill them as fast as a severe combined immunodeficiency disease, and it can be treated, so you kind of have to wonder, is it better not to treat them or better to treat them? They've also used gene therapies to specifically target cancer cells, so they can change killer T cells, which is an immune cell, so that they will attack cancer cells. Again, this is still in progress. They've tried it in the treatment of HIV, once again still in progress, and in the treatment of color blindness, which has seen some slight success. But to date, gene therapy has not been approved by the FDA in the US or Canadian agencies for use. It's still only in trial. And that's because there's many difficulties facing it still. So the first is there's problems getting stable integration of DNA. So we can get it into the right cells, but it's not integrating in a permanent manner, or if it is, it's not integrating into correct locations. So sometimes it's disrupting the DNA of the patient rather than curing it or fixing it. The other problem is it elicits an immune response. The vectors cause an immune response. So the retroviral vectors. And there was a case of an 18-year-old boy who was being treated as trial treatment and died because his organs shut down after his immune system over-responded to the retroviral vector. So today, the trials have been not very successful. We've got a long way to go before this becomes a common approach in treating diseases. And the final problem is that many diseases are multi-gene. Diseases. So it's not as simple to fix. So while there's promise, there is also work to be done. In fact, many of you are just in the right age group to be picking up this type of work as a scientist if it's something that interests you. It's a field that has a lot of room for growth and a lot of promise. Okay, we're just going to stop it there. And if you have any questions, you can come see me in class. Otherwise, I hope you have a good day.